Please join me in welcoming Dr. Kay Tye.
Hi, Jennifer, and hi, everyone. It's
Thank you
such
so
an honor
much.
to be here. Shall I just get started?
Sounds good.
Some stimuli carry innate emotional valence. No training or learning is required to have emotional responses to them. There's been a lot of controversy in my field about whether emotion is experienced by humans and animals in the same way. Of course, there's no way to ever know how an animal subjectively experiences emotion. There's no way to say whether it is the same as a human's experience. But there's also no way for me to ever truly know whether another human's emotions are subjectively experienced the same way that mine are. So even though I'm interested in emotion and how social stimuli can powerfully influence emotion, what I actually study is motivated behaviors. So with that disclaimer aside, I want to focus on the central question. Many of the most successful species on this planet live in social groups. Nearly all social species form hierarchies and find social isolation aversive. So how do we as individuals know our place within a social group? How do social groups maintain stability and structure even when there's changes in the internal composition or the environmental conditions? Finally, how do we adapt to changes in our social environments, both when they are under our control and especially when they are not? I don't need to tell you that our social environments changed dramatically between 2019 and 2020. In 2020, we witnessed that the rates of homicides and violent crimes spiked in a time-locked fashion with pandemic-related events. Anxiety and depression screens skyrocketed, with 70% of those individuals citing social isolation as the primary cause. Social isolation, or even just perceived loneliness, correlates with a shortened lifespan, mood disorders, increased mortality and morbidity for both cancer and heart disease, as well as increased inflammation. There is an urgent need to understand how these neuroadaptations are precipitated by social isolation. We do not know the mechanism of how social isolation or perceived loneliness causes all of these negative consequences. So there's an urgent need to create biologically grounded, mechanistic, predictive dynamical models for understanding this concept of social homeostasis, which will be the key to having a holistic and complete understanding of how social isolation precipitates the negative health consequences that it does. But before I talk about social homeostasis, which is this novel concept, let's just review the critical nodes within any homeostatic system. And what does even homeostasis mean? So homeostasis comes from the Greek words for same and steady, and just refers to any process that organisms use to actively maintain stable conditions necessary for survival. So there's been a lot of beautiful work done dissecting the neural circuits underlying energy balance, thermoregulation, osmoregulation, and I'll just give an example. So in a toy example, you can imagine thermoregulation. Detectors in my body detect that my body temperature is 100 degrees. It'll compare this to, it'll send this information to a control center, which compares this input, this detected input, to a given set point, probably close to 98.6 degrees. If there's a surplus or a deficit, in this case, there's a surplus, this information will be fed forward to the effector system, which are muscles, glands, anything. So that could be me moving into the shade, that could be sweating. Then these behavioral and physiological changes that are our response to the environment corrects for this change. And then if my body temperature comes back to 98.6 degrees, it'll shut down the homeostat and works just like a thermostat. Okay, so the notion of social homeostasis came to me through a serendipitous finding made by a former postdoc in my lab, Jillian Matthews. So what Jillian found in a study of dopamine neurons. These are these very understudied dopamine neurons in the dorsal raphe nucleus. So you probably hear all the time about dopamine neurons and them being reward juice and stuff like this. This is not the type of dopamine neurons that we're talking about here. There are many different types of dopamine neurons in the brain, including these ones nestled within the dorsal raphe nucleus. What we found is that just 24 hours of isolation in a mouse produces an increase in synaptic strength onto these neurons, and that after just 24 hours of isolation, we can look at the activity of this specific cell type and see an increase to a social stimulus after isolation as opposed to a novel object or to animals that are group housed. If we use a modern neuroscience tool called optogenetics, which is just genetically encodable control over neurons, which allow us to activate specific neurons in any pattern that we want. If we activate these neurons using light, flash light, and then using this technology, we can control neurons to have any pattern that we deliver. And what we found is that when we activate these neurons, we get increases in sociability, which is similar to other dopamine neurons, but it's actually an aversive signal. And so this was really confusing, because other dopamine neurons, they're pro-social, but they're also very rewarding. So what does it mean to have a pro-social signal that's aversive? And so I guess I would liken this to, use an analogy from our other research. So basically, if you think about feeding, this is just an analogy. You have food homeostasis, and I can eat for multiple reasons. I can eat because this dessert at the end of dinner is just delicious and I love it. I don't really need it, but it's delicious. So I'm eating it. Or I can eat this nasty fiber bar at the bottom of my backpack when I'm really desperate because I'm hungry and I want to escape the unpleasant state of being hungry. So there are both positive and negative emotional valences or motivational drives that can drive us to eat. Similarly, I may engage in social interaction and seeking social contact because I just enjoy it for social reward. Or I could seek social contact to escape the unpleasant state of being lonely. And so one thing we also found is that if we silence these neurons, normally it doesn't do anything. But after isolation, it blocks an isolation-induced rebound of social interaction. And I'll come back to that later. So this is something that we see. So let me just pause and talk really quickly about loneliness. The psychological definition of loneliness has 4 criteria. And I also want to say that loneliness is not a term that was previously used in neuroscience. This is a term from psychology, and it's defined largely by the following. 1, increased motivation to seek social contact. 2, it is aversive. It is an unpleasant state that individuals do not enjoy. 3, it is distinct from generalized anxiety. And then 4, a really tricky one, it's subjective. And that just means you can feel lonely at a party or in a crowded New York City street, or you can feel fine and not alone at all in your room by yourself. The point I want to make is that it is about how it is perceived. Leaving a party feels very different from being kicked out of one. Okay, so how do we get at subjectivity in a mouse? It's very difficult. There's no way to actually do this, but all of our mice are housed in identical laboratory conditions on a similar genetically inbred background, but within each cage, each group of mice forms a social hierarchy and each mouse has a different rank. The subjective experiences of a dominant and a subordinate mouse are likely to be very different, given that a dominant mouse has prime access to resources and mates with other mice performing affiliative behaviors. They might enjoy their social environment more than a subordinate that is being denied prime access to resources and mates and subjected to violence and aggression. So indeed, what we found was that the social rank of the animal strongly correlated with the degree to which all 3 of the optogenetic manipulations altered behavior that we performed. So this just suggests that there is this population of neurons could be the key to understanding the subjective state. But, how does this relate to social homeostasis? Well, by definition, if there is a cellular substrate for an unpleasant need state that drives motivation to correct it, such as hunger, thirst, or loneliness, there must be an upstream system to detect that deficit. And so, preliminarily in our working model, we are thinking of this population of neurons as representing an effector system for social homeostasis. Brief aside, some of you might be thinking to yourself, like, who cares about mice? What about people? Lots of, how do we know that the circuitry would be even common at all? Like, is there any point? And for this reason, in collaboration with our colleagues at MIT, we reproduced this effect in humans. So this experiment, this study was just published in 2020. In humans, we're only allowed to socially isolate people for 10 hours because longer than that is considered torture. But within 10 hours, just 10 hours of social isolation, we also observed an increase in the same brain region using a different signal, bold fMRI, in response to social stimuli. So of course, it's not the same resolution, but as much as we could do in a human, we've reproduced this effect. And that's very exciting. That suggests that the findings that we have here will be relevant for humans. Okay, so now that we have identified an entry point of an effector system, an effector system must receive input from a control center that receives input from a detector that senses the quality and quantity of social contact. And this is high-level sensory processing involving association and memory systems. But another component that isn't in other homeostatic systems that I think must be necessary in a social homeostatic system is some recognition and identification of the individual's social rank that can suppress the effector system and or modulate the control center. For example, say I'm a subordinate male within a hierarchy, and even if I want to mingle with the dominant males or mate with these females, I might suppress this desire to avoid punishment from the alpha. This rank decoding node must receive input from the detector and also provide feedback to the detector. And then, of course, like any homeostatic system, this one requires feedback from the environment. But this is non-trivial. Feedback in the environment, if we're talking about thermoregulation, you just measure the temperature. That's not the case. Measuring the interaction with the social group is a non-trivial measurement, and we actually had to do, this is 5 years of tool creation and tool development to give you this. So what I present to you now is Alpha Tracker. This is something that we've been working on for a while, and it's very necessary. So in addition to developing new tasks wherein we can record wirelessly, I'll tell you about that later, we created a deep learning computer vision platform for multi-animal tracking that does both pose estimation and behavioral motif discovery. So additionally, we have incorporated an unsupervised clustering feature to facilitate unbiased discovery. So what does that mean? That's a lot of jargon of sort of artificial intelligence or deep learning. But what we're able to do is not only track the different animals, but look at their different interactions and discover what they are. Even if experimenters didn't say, oh, when they do this specific behavior, this little sequence of behaviors, that means this. Maybe we didn't know what to look for. We don't have to know what to look for. We can let math tell us what has changed in the behaviors. And I think this is really powerful because it allows us an unbiased way to discover what is actually happening. Importantly, this tool has been pre-printed and is freely available on our GitHub, no strings attached. Download if you want to. Okay, so, So how do we dissect this circuit? Coming back to this thing, I've mapped out the nodes that we absolutely need. There could be other ones as well. But I want to first say a couple of things. I'm not one of those people that thinks that one brain region does one thing. I think it's more likely that each of these processes will actually be implemented by distributed circuits that lie, across the brain. But for simplicity's sake, when tackling a big question, I hope you'll bear with me. So this next slide is an oversimplification. But let's break this down. So now that we have a putative effector system capable of driving the constellation of features that comprise a loneliness-like state, we can explore how this effector system implements each facet. And what do I mean by that? So, I described loneliness as having multiple components, including there's the aversive component, there's the prosocial component, there's other components. And so what we found is that dorsal raphe nucleus dopamine neurons that go to one brain region called the basolateral amygdala posterior are important for the avoidance or the aversive components of this behavior that is elicited. You can see that here, animals will avoid the side of the chamber that is paired with photostimulation. Not true for the other outgoing projections. A different projection of these same dorsal raphe nucleus dopamine terminals, but terminating in the central amygdala, drives sociability in a social rank dependent manner, but not aversion. So these are separable. These constellation of features are actually driven by different circuits. Okay, so this is really interesting. The central amygdala might, inputs to the central amygdala drive sociability. So what's going on there? So to probe how dorsal raphe nucleus dopamine neuron input can influence the representation of the neural representations of social stimuli within the central amygdala, my graduate student Christopher Lee simultaneously imaged using a genetically encodable calcium indicator that helps us visualize neural activity. So these cells will light up whenever they're active. We can look at this in the central amygdala while stimulating dorsal raphe nucleus dopamine inputs. We can run animals in a task where they can explore a space that either has one side that has a social agent and another side of the chamber that is empty, and then look at what all of these neurons are doing. And so we look, taking a global view of what these neurons are doing. We can zoom in here. There are these two different populations of neurons that respond totally differently to juvenile input, depending on whether the light is on or off. So given that stimulation, turning on the light and stimulating DRN dopamine neurons can mimic a loneliness-like state, this suggests that they might be different ensembles of neurons, different populations of neurons that respond to social stimuli when we are lonely and when we are not. So DRN dopamine neurons contribute to the effector system, and any putative effector system needs a control center. So how do we find this control center? Well, we can use monosynaptic rabies tracing to identify upstream input to the dorsal raphe nucleus dopamine neurons and create a short list for candidates of the social homeostatic circuit. And so we've done this. And in addition to the lateral hypothalamus providing monosynaptic input to the dorsal raphe nucleus dopamine neurons, meaning it directly plugs into these cells, my very first graduate student, Edward Nye, found that stimulating lateral hypothalamic GABA neurons could drive increased hypersocial behavior. And this is one reason that we looked at this. We suspect that circuit loops, including the anterior cingulate cortex and basolateral amygdala, may represent the detector system. Insight towards this node was provided by the thesis of my former trainee, Stephen Azier-Alsop, who is now faculty at Yale. Here's some examples of the behavioral paradigm. So in this task, what we were doing is to look at how an animal learns about the world by watching another animal. And so in this task, an animal would receive a single shock, then be moved to the other side of the chamber where a cue would predict shocks to another animal. So this pairing was never experienced by the observer animal, and only is being experienced by the demonstrator. So I'll just show you a movie because it'll probably make it easier to understand. So here I'm showing you a clip. On one side of the chamber, there is a grid floor where the animals can get shocked. And the other side of the chamber, there's a solid floor. So that time there was a cue presented, and you can see even though the animal who's not going to get shocked isn't going to get shocked, they still freeze because they're learning to freeze by watching the other animal. This movie is a little bit more clear. Okay, this is mimicking. So keep your eye on the animal on the solid floor that's closest to us. They're freezing to the cue, and now watch what happens. So the other animal is about to get shocked. Boom, they're getting shocked. Now this animal also is not getting shocked, but also mimics this jumping behavior. And so this is the phenomenon that we wanted to capture. How does an individual look at another and then extract whether this is good news or bad news? Is this something that we should be afraid of? How is this being processed? And so we looked at the anterior cingulate cortex and basolateral amygdala, which in humans has been shown to be important. And also in mice, we've shown that from lesions, we know that the circuit is important for observational fear learning. Sorry. But what we don't know is the directionality of information. Where is information coming in and being sent, and what information is actually being processed. And so this is a little bit complicated, but I'll try to explain it in a way that you can understand. So the idea is, so here I'm showing you a peri-event raster histogram. Oh, maybe you can't see my mouse, but here. I'm showing you a peri-event raster histogram, which each row is a trial. Each dot is a action potential, a spike being fired by your neuron. And then all of those trials are synced up to this cue. So basically, every time the cue plays, we're looking at the neural activity that happens. So it's like, okay, maybe I don't care the first time because I don't know what this cue means. But then once we start the conditioning, when cues are being paired by shocks and the demonstrator is getting shocked, suddenly now there's something to learn. So what we wanted to ask was, who learns first? Is it the amygdala or the anterior cingulate cortex? And the way that we asked this was using a state space analysis. So if you allow me to show you how this works, basically what we're doing is we're taking this response window right after the cue is being presented and asking, how much did this neuron fire in response to that cue on each time the cue was played? So the cue is just a little tone, it goes beep, and it says, I'm going to deliver a shock to the grid side of this floor. So what we can do is take each of these trials and then transpose them. So you can see here, the trials are moving downwards, here they're moving across, and we can see, okay, when we're just habituating, we don't see anything, but then right here, this is the learning trial right here. This is the rate change trial. So when would you say that the neuron has learned or changed its activity? And using this analysis, we would determine it's here. So this is really quick, just a couple of trials. Initially, we were like, wow, this is amazing. It means the anterior cingulate cortex is learning super fast. And if we look across many different neurons, here the anterior cingulate cortex is plotted in orange, and the basolateral amygdala is plotted in these open bars, overlaid. And what we're looking is a number of neurons and how fast they learn. So in general, anterior cingulate cortex neurons were learning a lot faster than basolateral amygdala neurons. And right here is the first conditioning trial when there's something to learn. So, okay, we thought when we first did this analysis, oh, this means the anterior cingulate cortex is learning first and then teaching the BLA, the basolateral amygdala. But this was only because we were looking at the neural activity with cue onset. We needed to look at more. Okay, so we did another analysis, the same task, again, looking at habituation and then conditioning. And let me just orient you to the next slide. So what I'm about to show you is called neural trajectory analysis. So what I'm going to show you is just a little plot that is capturing the high dimensionality information of the entire ensemble, hundreds of neurons that we're recording. And how do we depict that? How do I show you what hundreds of neurons are doing at the same time? I can reduce the dimensionality and then make it this abstract drawing. Okay, so I'm going to just show you that. And just to orient you to the next slide, each trial, it's averaged, and it's going to begin with the circle. That's the baseline. So that's before the cue is played. So we're getting a little bit of baseline. Then the cue will be played at the triangle, and then we end the trajectory before, the shocks are delivered. Okay, so what this looks like is first, what we're doing here, once again, is we're trying to basically plot hundreds of neurons activity into dimensionality reduced space. So don't worry about that precisely. Just think of this as an abstraction. So what I'm showing you here is that there's some animals that were in the paired group. That means they were watching an animal received paired presentations of the cue and shock. So they have something to learn. In the other case, the control group, the unpaired group here on the right, there's no pairing. There's cues and there's shocks, same number of cues and shocks, but they're not related. So the cue doesn't predict when the shock will be delivered. Okay, so what we saw here was pretty striking. Only the basolateral amygdala actually learned what the cue meant. Here, what we're seeing in all the other groups, yes, there's movement. Habituation trials are different than the conditioning trials, but the shape is basically the same. In all three of these other quadrants, the shape basically stays the same. You're just moving it over. It's just a transposition. And that makes sense. The presence of the demonstrator, the mouse that's getting shocked, being there, the animal being in distress, could change the environment of and the representation for the observer, but only in the amygdala is the meaning of the cue actually being learned. Okay. So what we're suggesting here is that the anterior cingulate cortex, this brain region here, it's changing very quickly because it's rapidly expressing state changes, such as demonstrator distress. Only the basolateral amygdala actually learns through observation. So information that is extracted from the social agent is being fed into the anterior cingulate cortex and then put forward to the basolateral amygdala. And of course, we did a lot of other experiments that I don't have time to talk about today. But this work was done by Anthony Burgos Robles and Jarong Chung. So if you're interested in this, there's a lot more of other experiments, but it's published. So I'm going to move on to the unpublished stuff. So this final project I want to tell you about is my current postdoc, but soon to be faculty, Nancy Padilla-Coriano. And the idea of this project was to simulate this following scenario. So, I mean, this might not happen for all of you, but in pre-pandemic times, when there would be a seminar, a scientific seminar, often there's pizza. And if you arrive late to these seminars, there's no more pizza. So say you get to the seminar 3 minutes late and there's one piece of pizza left and it's you versus your boss. What are you going to do? You're kind of hungry, but your boss looks like they want this pizza too. So this is the scenario, and it happens all the time. You're at dinner, you're being polite. Do you want that last piece of food or not? What's the deal? So we wanted to mimic this in animals. And so basically we have a task that's called the reward computation task. And we had to create this task so that we had a way that there was to investigate this and also to create statistical comparison that could be rigorous. So first, we established ground truth of what the animals' ranks are using a well-established task called the tube test. The tube test is great for reliably telling us social ranks. It's based on the idea that mice live in burrows, and sometimes burrows can be very narrow. So you can think about it like a hallway. If you're walking down a narrow hallway and there's not very much space, if you see someone else coming, you either walk forward and you expect them to wait for you, or you back up and make space for them and yield to let them pass, and then you can go after them. But which one do you do? So it's going to depend on, your relative rank. And in mice, it's very reliable. The dominant animal gets to walk. The subordinate animal will back up and wait. The only problem with this task is that it doesn't allow us the granularity to make some of these finer comparisons because, if we see neurons responding during the tube test, it could be because of their dominant, but it also could be for a number of other things. It could be that they're pushing. It could be that they're getting pushed. We don't, it's hard to control what's happening and figure out what the neurons are actually responding to when so many different things are happening at the same time. So what we did is we train animals to learn, that this cue predicts a sucrose delivery. It's like a little chocolate milkshake thing. So it's just saying, hey, I'm teaching you that this cue signals food. Okay. That might be like learning where the table is, where the pizzas go at seminar. Everybody can learn this the same. Doesn't matter what your rank is. You can figure out that this cue predicts food. That's not difficult. Then we do a round robin. We do a pairwise comparison of each pair of animals. So the rank 1 animal will battle it out with 2, 3, and 4. Rank 2 will battle it out with 1, 3, and 4, et cetera, et cetera. So every animal will battle it out, will match up against each other animal. And what we show is that, yes, even though dominance will dominate, they will win more rewards, they will push more with greater success, they will be displaced less frequently, and they'll just get more rewards. They don't win all the time, and that's good. That helps us to understand how wins and losses are processed differently or similarly by subordinates and dominants. So to do this, we had to overcome a lot of technical challenges. We had to be able to record neural activity wirelessly while identifying specific populations of neurons. And we had to design this. So this is what this task looks like in an earlier version of this task. These little hats are smaller now. The recording devices are a little bit lower. But Nancy Badia, along with Javier Weddington, now at Stanford, and Mackenzie Paterino, now at University of Washington, helped develop. So why are we looking at the prefrontal cortex? Well, we know that in humans, medial prefrontal cortex is very important with social rank learning in humans. And we also know that optogenetic induction of plasticity in the PFC can actually change social rank in mice. The prefrontal cortex is conserved to different degrees across species. And we just covered this in a book chapter. The first author is Austin Coley, a postdoc in my lab. And if you're interested, check it out. Okay, so really our question is, we know the PFC is important, but what in the world are these neural ensembles actually doing? You know, prefrontal cortical neurons have complex computational capabilities. So I'll explain this neural trajectories idea one more time because it's pretty important this time. So it's the same idea, but again, if we record from, let's say, 1,000 neurons, and I want to show you everything about what's going on with those 1,000 neurons, it's really hard. Like one way I could do it is plot the trajectory across time because, you know, all the neurons are changing activity at every millisecond, the activity is different. And there's 1,000 moving pieces. So maybe if we had 1,000 dimensions, I could plot something in 1,000 dimensional space, and then you could visualize that. But of course, it's really hard to plot something in 1,000 dimensional space. And so what we can do is reduce the dimensionality and collapse a lot of it, because a lot of the variance that you see is redundant. A lot of things are moving in the same direction because, and that covariance can be captured. So that's what I'm going to show you on this next slide once again. So on the left is an example trial, and on the right is trial average. There's going to be 4 different trajectories. There's going to be a win trajectory and a loss trajectory for the trials that mice are either winning or losing, and I will give you those wins and losses differently for subordinates and dominants, depending on in that particular session if they were subordinate or dominant. So this holds, and this is true regardless across different individual animals, I should say. So what I'm showing you here is, okay, there's some baseline activity, right? Boom, now the tone just went on, the cue went on, and you could see these trajectories kind of accelerate. All right, so what I want to point out to you in these trajectories, so you're going to see 4 lines. What is notable to me is that they all start in different places and end in different places. They each kind of occupy their own corners of activity space within the brain. And so essentially the pattern that we found over and over again is that there are different neural activity spaces with the self and the other. So we have some representation for when we are doing stuff, when we are getting the reward, or when we are moving over here, versus when the other animal is getting the reward, or the other animal is moving into that particular space. And then we have different representations for winning and losing. So subordinates and dominants occupy different spaces, and wins and losses occupy different spaces. So, okay, that's great. That's kind of abstract, okay, that's cool. And it suggests that we'll be able to predict some of this information later. But can we, it's kind of hand wavy. What if we could actually predict, the very specific behaviors of the animals using a model? That would suggest that we really understand what's going on in the brain. Okay, so that's what we tried to do next. So Kanha Batra in my lab, this is pretty cutting edge, I would say, machine learning approaches. We are using, for aficionados out there, we are using a hidden Markov model combined with multiple generalized linear models. But what this really just means is that we are taking all the neural activity from the prefrontal cortex. We're using Alpha Tracker to discover different behavioral states. And then we are asking, can we predict what each animal is going to do at any given moment based on just only with their neural activity? And we were able to do this much better than other models. So what I'm going to show you this time is an example trial at the top of the video of the mice. On the left, I'm going to show you sort of just the raw data that we're getting from the medial prefrontal cortex, which isn't really like looking like anything, like, just bare eye without any analysis, without any computational power. I don't really see anything. But when we put it through our model, or compared to a bunch of different models, you can see how well it predicts. So what I'm going to show you here is a number of different models: a generalized linear model, a support vector machine model. These are just comparisons. Our model, the predicted behavior from our model and then the behavior. So here goes. Here's the video. So this is the actual behavior and this is our predicted model, okay? And you can see here that our predicted model does extremely well in terms of accuracy of predicting the behavioral states. And if we quantify this, comparing it to other models, you can see it does extraordinarily well, predicting like in the 90% accuracy exactly what the animal is doing, just based on the neural activity from one brain region. Okay, so to us, this was pretty astounding. If prefrontal cortical neurons truly represent social rank, then we should be able to decode social rank from PFC data PFC data alone, the neural recordings alone. But when is it going to be encoded? Like, am I always thinking about my social rank? Am I like day in and day out? Am I constantly thinking about my social rank? You know, if I'm in a room with my boss, am I constantly thinking, oh my God, they're my boss and they outrank me? Or do I only think about it when there's a competition, when there's a conflict? Like when it's time for us to, we both want the same thing. That's when, you know, they pull rank on me, quote, so to speak. Maybe that's how it works. How does it work? I don't know. Or we didn't know, now we know. So I'm going to tell you. We used a support vector machine classifier to test the decoding accuracy. So if you're interested in how we did this, I'm happy to tell you, but basically the same thing. We did dimensionality reduction on the neural activity and then trained the model on some of the data and then tested it on data that we didn't train it on. And what we found was that PFC neurons significantly decode both competitive success and social rank. So for competitive success, I mean, this is not necessarily a given because, you know, wins and losses, like anyone could win on a given trial. And we've controlled for the fact we've done trial matching to account for the fact that, you know, animals might win different amounts of trials. So we controlled for that. And I guess it's not surprising that we see such stable recording of relative rank. But what we found that's really interesting, I'm not showing you here, but you can see that you can decode social rank. You could decode even who's going to win and lose the next trial before that trial begins. That's like saying I could before the contestants have stepped into the ring, I can tell you who the winner is going to be just by listening to their brain, by one of the animal's brain. You can predict if they're going to win or not. And this is really exciting. We can do this for up to 30 seconds before the trial begins, which is as far out as we're capable of looking based on our experimental design, but there's no limit to that. So what we think, what I would interpret this as is we have found cellular resolution evidence for this phenomenon, this hand-wavy phenomenon called the winning mindset. You can actually predict. It is predictive of the subsequent outcome even before the trial has begun. And I think that is pretty exciting. Okay, so now we want to figure out prefrontal cortical neurons go to many different brain regions. They're all over. And we wanted to think about like which ones were important. So to determine how subsets of cells contributed to social rank encoding, we played with how including different subsets into our support vector machine classifier, our decoder, impacted decoding accuracy. So basically, we found that prefrontal cortical neurons that project to the lateral hypothalamus, this brain region I just told you about, and that is important for social, like hypersocial behavior, contributed more heavily to social rank decoding than randomly selected cells. While PFC neurons projecting to the BLA, which I told you was important for learning, wasn't actually important for social rank. We then found if we caught, so that's nice, there's this correlation, but we want to like really prove it. So experimentally, if we now manipulate this projection from the medial prefrontal cortex to the lateral hypothalamus, we can promote dominance behavior. And when we activate these neurons, these animals will occupy the port more, they will win more, they will push more, they will get more rewards. And we did not see any differences in locomotion, anxiety, or effort. Okay, so this is just an overview of how we have begun to build out a skeletal understanding of social homeostasis. Finally, we have established the enabling technologies, and we've only just begun accelerating down the jetway. And like, I'm really excited to see liftoff of this field in the very near future. But with this foundation in place, and also in the backdrop of everything that's been going on in the world in the past 18 months, we can begin to ask questions that explore other dimensions and timescales. which brings me to the second big question of my research program, which I only will talk about very, very briefly because it's forward looking. But what changes from acute to chronic? So after acute social isolation, there's when individuals are reintroduced to their social group, there is a rebound of social interaction. So, you went on a trip, mommy's back, yay, welcome home, that kind of pro-social rebound of interaction, right? Family reunions, stay up all night and catch up, versus chronic social isolation, which we've seen, through the pandemic, through families being separated, other situations like this. With chronic isolation, when individuals are reintroduced to their social group, they are territorial, they are aggressive, they are avoidant. There is an anti-social response, which is really baffling from a scientist's perspective, because we've got social isolation, the same manipulation, and then the same stimulus, reintroduction to the social group. And then you have these opposite responses that are either pro-social or antisocial, that are positive valence or negative valence. Like, how does this happen just across time? So in our model, what I think can often happen is, sorry, my mouse is out of control, but here we are. This is the orange line is what you're actually experiencing. The black line is your homeostatic set point. And you're trucking along. Oh, there's some social isolation event. Okay, now a deficit is detected. My effector system will be activated. I'm going to start exerting, exerting more resources towards seeking social contact and maintaining my previous set point. And then, maybe I call a bunch of friends, go out and find everybody and have host a party at my house. I don't know, do some activities, and I can bring this back to my set point. Great. Now, the quality and quantity of social contact is at my set point. Everything's all good. And with this acute isolation, you can see this pro-social rebound. But what about when that isn't possible, when we're not in control of our social environments, when there's chronic isolation that we can't stop? Perhaps there's government mandated lockdowns and quarantines. I don't know, social distancing, any of these possibilities. There's social isolation. I detect this. My effector system is activated. And then, there will be an increased effector system drive. There will be more effort to correct this deficit. And so maybe across some amount of time, or maybe it's the amount of effort that is expended, animals, individuals, humans and animals alike, will eventually give up. At some point, I'll stop calling. At some point, I'll stop inviting people to parties if they don't come. There's some point where the behavior of seeking social contact stops. And this is where we hypothesize that there is a homeostatic set point adaptation, which is adaptive. We come down to the new normal. Okay, now we're adjusted to our new normal. After which, if we were then reintroduced to the social group, it might feel like a surplus, right? If we were now brought back up to our previous optimum would now feel like too much now that we've adjusted our set point. So this is something that we want to investigate. Okay, so what I've shown you today is just a high-level conceptual model on the very tip of the iceberg. In the coming years, I aim to develop these predictive dynamical models, and I just want to stress that understanding the neural mechanisms and processes that allow us to maintain stable hierarchies and undergo social homeostasis is going to be key to understanding the core of many different mental health disorders. Social isolation is a cause of many different mental health disorders, and is also any impairment in social interaction is also a symptom, a key hallmark feature of almost every psychiatric disease state. So this is a basic science question, but understanding it could not only help us understand how we are the way we are, it could also help us potentially solve the mental health crisis. With that, I'd like to thank everyone on my team, staff, graduate students, postdocs, volunteers have worked really hard on this research program. I thank my collaborators, my funding sources, and you for your attention.